This was important. What we're going to do is we are going to degrade our 20 amino acids. But what we're going to do is we're going to degrade them to a point of getting the carbons into a form of a pathway that you've already seen. Okay, so I'm not going to take them all the way down to CO2 and water, but I will get it to, let's say, pyruvate. And then once you're in pyruvate, you know in the degradative process, it will go through the citric acid cycle and be degraded to carbon dioxide. Or I will get it to a citric acid cycle intermediate. Once we're there, you know how to degrade from that point in time. And so if you take a look at the number of amino acids, there are 20, right, in those boxes? If you count them all up, there are more than 20. But we only have 20 amino acids. How in the world is that work? Someone's lying to you, right? Yes, there's some that enter in multiple places. The ones that are very large or have a lot of carbons can actually degrade into pieces that enter into different cycles. So if you've got glycine, that's one little thing, it goes one little place, right? But if you have tryptophan, which is huge, those carbons can enter in the cycle in different places, all right? And so don't let that confuse you. You may have different entrance points for the same amino acid. Now, our book liked to group the amino acid degradation into, I think they actually did 4 groups. I actually identified 5. And then we've got a lone leucine who does his own little thing. All right, and so we can have and identify 19 of the 20 amino acids in some sort of pathway that are grouped together, 1, 2, 3, 4, 5, plus leucine. All right, if I look at group 1, all right, what we see is we have threonine, we have glycine, serine, alanine, cysteine. They all seem really tiny, except for tryptophan. Tryptophan, part of the reactions will create alanine, so that's how tryptophan will interact here. And then many reactions later, we got something else. And so some of these reactions are really specific. What I want to do is identify for you the pathway for threonine. And so we're going to take a look. Threonine will break down into 2 products in terms of the carbon chain, an acetyl-CoA, and we will break down into a glycine, which then can be further degraded. And so acetyl-CoA, what pathway are we in? Right before the citric acid cycle. Okay. The threonine's reaction has a couple that I'll articulate and a couple steps that I will not. I can articulate the first step, okay? And in that, what we're going to do is we are going to have a oxidative reductive reaction. Now, I didn't draw something. What do I need to articulate on the drawing for this reaction to occur, where I have an NAD plus as the reactant and an NADH as the product? What don't you articulate sometimes on organic structures? It's there, you just don't see it. A hydrogen. And so if I look at 4 hydrogens, I have one here, I have one there. That just isn't articulated. All right, in this, the mechanism would be such that I take this proton, okay, I will take the electrons to form the carbonyl, and the leaving group would be a hydride, and I can only do that because of hydride acceptor, and I've created the ketone. So I took the alcohol of the R group of threonine and created this particular ketone. All right. all well and good. Now, this ketone here is very similar to what we did with the reactions that involved the amino acids. And the amino acids had a PLP as part of the reaction. Now, I'm not going to do that mechanism because we have done this. A PLP is this organic structure. And what we're going to do is we're going to react this ketone with the nitrogen of the PLP, and I'm going to create that Schiff's base. So instead of double bonded oxygen, I get double bonded nitrogen. This is the PLP. So I didn't articulate that step. All right. Our next reaction is acetyl-CoA. Acetyl-CoA comes in. Oh, sorry, not acetyl-CoA, but a CoA. Deprotonates the proton. I will attack it at the ketone, and then I'm going to show it kick up onto the oxygen. And I'll draw an intermediate for you. Okay, because the next step is not exactly what you might expect. Okay? And so, in that intermediate, when I kick up, I have a couple options. I can kick up, pick up a proton. I would pick up a proton if in the product I would have a hydroxide. I do not see a hydroxide. And so, if I kick up and I don't pick up a proton to become a hydroxide, then I must kick down. I will kick down, okay? And what I'm going to have is a leaving group. Now, the leaving group is facilitated because I have the cofactor in. All right. Now, what I've actually formed at this time is the CoA, a ketone, and a methyl group. This is the acetyl-CoA that comes off. Now, because I have the cofactor, watch where the bond breakage, which is this bond, goes. It will flow here to form that double bond. There's no double bond here, so this one moves here. And do you see something different between the ring and here? Once that moves, I've got to move these pairs of electrons, and this one will go here, and this one will go there to relieve the positive charge. So I have an acetyl-CoA fall off, and then I have all of these double bonds in conjugated system move around to where I have a lone pair of electrons sitting right there. And that's a pretty good leaving group because I went with a charged, what would be a charged leaving group into a neutral leaving group. All right, the CoA has come off. When you look at the original, okay, you just knocked off this piece right here. The only thing you have left is an acid, this carbon, and that nitrogen. Well, here's that acid, okay, there's that carbon, and there's that nitrogen. All right, that stabilizes the transition state, but I can make this re-aromatic by putting the electrons back, putting the electrons back into the ring, and now picking up a proton. And you can see between this one and that one, that was the only change in terms of the hydrogen. But you can see you reformed the aromatic ring. Going from this to here, what is the difference? Okay. The difference is I have two hydrogens here, one there, one hydrogen there, one there. I'll take off one of those. The electrons will form the double bond, and this will pick up a proton. The original PLP was bound to a nitrogen through a Schiff's base. I've reformed that. I can now have the enzyme come in, and just like the aminotransferase attack, and then this falls off. Well, what is this that falls off? Glycine. And so threonine, if you go back to the past slide, breaks off as acetyl-CoA and glycine. It is facilitated by the PLP cofactor. All right. Group 2, I'm going to articulate a lot more of these reactions. And so we have histidine, arginine, proline, glutamine, and then glutamate. All right. And we know glutamate, we've seen the reaction that changes glutamate to alpha-ketoglutarate. That's the transaminase, or the, GD, that's really good. Glutamate dehydrogenase can take the glutamine and create alpha-ketoglutarate. That is the one step before the urea cycle. And once we have alpha-ketoglutarate, we're in the citric acid cycle. So these are related by glutamine all goes to this, proline will put its carbons there, so will arginine has one way at which it puts its carbons there, and histidine. So they all put their carbons into alpha-ketoglutarate to be broken down. We will do them in different pathways. Now, I don't have to show you this pathway here, where it goes ornithine and down, because that's part of the urea cycle, and we've looked at that reaction. Just to re-emphasize, we looked at this before in this chapter, where glutamate creates a Schiff's base, water react with the Schiff's base to keep alpha-ketoglutarate and the ammonium coming off. This will go to the urea cycle. All right. Here is the breakdown of proline. All right. We have a reactive or a oxidative reductive reaction, so we have the FAD as an acceptor. All right. So we got to identify where in the world. All right. Here's proline. Acid, acid, hydrogen, hydrogen, carbon, carbon. Ooh, 2 hydrogens, 1 hydrogen. Ooh, a double bond. No double bond. 1 proton, 1 proton, or 2 protons. So it looks like we're doing reaction right about here. All right. The most acidic acid in this of the two would be this one, because in the intermediate you could make this neutral, but those electrons will form the double bond, and the hydride will be accepted by the FAD. So I have the unsaturation. When I look at the next reaction, I have a hydroxide that's attached here. I have a single bond, and I have a nitrogen with 2 hydrogens. I have a double bond with a nitrogen and 1 hydrogen, and I have nothing attached here. But I react with water. So what am I going to do to the water? Deprotonate. And that becomes a good nucleophile because I have an OH. The OH attacks here. Okay? It's a double bond. It could attack one side or the other. Why does it attack at that carbon? If you were in an organic flask, it could attack both, and you probably would get both. One would be a major product, one would be a minor product. But what is different about an organic flask and a biochemical reactions? The enzyme orients only one attack. Also, when you attack that, the intermediate could be a lone pair on this nitrogen, which would be neutral. That's not a bad intermediate. But because of the pH of the system, the pKa's, that will pick up the proton to create the 2 protons. Okay? The next step is kind of a little bit different. Okay? And here is where you would look at that and go, those two things don't even look the same, do they? Okay? Here is that one. Okay? So, this one is a carbon that is bound to one hydrogen and bound to a nitrogen. And so, that's that one. And so, this right here, that's nitrogen, is that one. So, that nitrogen here was this one. It was bound to that carbon. Well, where in the world is that carbon in the product? It becomes the what? It becomes the carbonyl. So that's where it is. So now I know that the oxygen, which is a hydroxide, has to be a carbonyl. Well, I can make it a carbonyl by using a base, right? Take that off. The electrons form the double bond. And then I have to have a leaving group. The leaving group is the bond that was broken. That bond that is broken picks up and creates the amine. Glutamate gamma semialdehyde. In the big classification of all types of molecules, that is an amino acid. Not one of the 20, but it is an amino acid. All right. I will show you a little bit of the arginine because we actually looked at it. We actually didn't do the mechanism. All right. If I take a look at the reactant and the product, what in the world are we going to do? Anybody? If I take a look, is there any change here to here? Nope. All the change revolves around this area, right? And I have two nitrogens. I'm missing a double bond. Here's the carbon, so it must have a hydroxide attached to it. If I take the water, deprotonated, I have a hydroxide. And then I'm going to attack. And then this pair of electrons is just going to go onto the nitrogen to make it neutral. Okay. Yes? The enzyme is going to orient it. So if you're doing an analysis, you just need to see where the attack has to be. Now, where could that attack? Lots of different places. And if you just put it in an organic flask and you didn't have any direction, you'd have lots of reactions. But because it's in an enzyme, it orients those two reactants right next to each other. All right. What do you think is going to fall off in this reaction? What is the leaving group? I didn't write it. Yes, everything past it. And everything past it would look just like this. What is the name of that structure? What? It is an amine, but specifically. You told me it was not yellow last lecture. It's urea. Okay. So I've got to make urea, and I do that by taking that proton off, creating the double bond. Okay. And I will pick up one proton. Okay. Does that actually make what I drew in the at the end? Is that exactly what I drew? No. What would come off is NH2, right? But all amines that are floating around have a pKa of about 9 to 10. The pH of the solution is about 7. So by pKa, I'm going to pick up another proton just because it is so basic compared to the rest of the solution. So that is what that's why I get an extra proton. An aminotransferase, okay? Aminotransferase generally had this reaction. An aminotransferase would react, and I transfer it into a ketone. It was a big, long mechanism. It was a two-stepper. And so in this particular reaction, I have a hydrogen there. The aminotransferase is going to take that amine off and substitute it with a double bonded oxygen to get the glutamate gamma semialdehyde. All right. I looked at proline first, okay, because I ended up as glutamate gamma semialdehyde, and then I looked at arginine, and I'm at glutamate gamma semialdehyde. This point in time, the body doesn't know where it came from, either arginine or proline. The next step, we're going to get into something that we can degrade. And so you can see here, I have one hydrogen. I have a oxygen bound. I have an oxygen that's going to pick up a proton, and then I have to have a hydroxide bound there. Well, if I have water, I can always add a hydroxide by taking a base, deprotonating, attacking, and picking up a proton. All right, I'm showing you lots of mechanisms. That could have been a test question. I will give you the start, and I want to give you the end, and you will have to get from the beginning to the end. I will not make you do a 5-step reaction. I will give you a reaction that is much more defined. All right? The next reaction here would be from here to there, and I can see, I have a whole bunch of stuff here, and that looks a whole lot different there. All right? What I am missing on this carbon over here is a hydrogen. Ooh, I have an NAD+. I know I can have a hydride acceptor. A base will deprotonate a hydroxide. Do I have to attack the bottom one? No, because they're equivalent. So if I'm attacking the top or the bottom, it's not any different. I form a ketone, and I have a hydrogen that carries a pair of electrons called a hydride. Attack the NAD+ to accept it. My mechanism drew this. A double bonded. Okay, but my product has this form. Why does this turn into that without me showing you? The pH and the pKa. What are the pKa's of acids? Kind of somewhere around between 2 and 4-ish, really low. And the pH is 7. Definitely at least one full unit above it, so it's going to deprotonate it. Okay? Here, they can be fully protonated because there is no reason, the pKa's are not anywhere near. Only when you get into this form does the pKa shift down into that low pKa form. At this moment in time, glutamate, we know, can be degraded to alpha-ketoglutarate, where I showed you a couple of slides before. So we're at a place in which we know where degradation occurs. Histidine. Ooh, boy. This one's a little different because we've got that ring structure. All right? What in the world do you think happens in this first reaction? Okay? If you are inventorying, another thing that you probably will want to do is make sure you know where all your protons are. Okay. And since the ring is the same and nothing happens around it, that's okay. If I put where the protons are, does that help you out about what you need to do with that one? Okay. Another thing I could help you out is doing is having minus NH3. Okay. Actually, NH3 won't be in the body. It'll be NH4 plus. Again, a pKa issue. All right. I'm going to take a base. I'll take off the proton. I will form a double bond. And I'll pick up a proton. Can anybody classify what kind of reaction I just did? A what? It is an E2 mechanism, yes. Okay. Base comes in, deprotonates, form the elimination, and the leaving group leaves at the same time. All right. That one is straightforward. This one's fair game. This one is not fair game. Okay. It looks like you're going to add a oxygen here, right? And I could add a hydroxide right there, and then that could pick up a proton. But something weird's going on because I also have that going on. There's a loss of a double bond. So there's something else going on. This is not one I can do because the mechanism looks just like this. Yeah. Okay. What I want to show you is, we know what NAD plus does, right? NAD plus is a what? It's a hydride acceptor, right? Most of the time. This is a mechanism in which it doesn't accept a hydride. Actually, the first step in the reaction is the amino acid reacts with the NAD plus in a totally different way than what we had wanted it to do. It has ring cyclizations. It's got a lot going on. There's a water coming in eventually, but it's not the first step. And we will get to the end, but more complex than I want you to know. So this slide just pretended it didn't exist in terms of studying, but I just wanted to show you some reactions are very complex, and we don't want to deal with them because it's not something that you could have predicted. Okay. All right. In this case, this is a reaction that is open. Okay. And so, what in the world would you do with this one? Okay, I've been doing all sorts of mechanisms. Talk to your neighbor. What would you do with this particular reaction, and how do you get from the reactant to the product? Okay, do you feel good? Can you do it? Do you feel strong? Confident? Able to do it? Okay, are you feel confident in telling me what to do? Oh, come on now. You got the chance to tell what the professor what to do. Yes. What should I do? Okay, that's right. A base is going to take a proton off the water. Now I have a pair of electrons. Someone else, what do I do with that pair of electrons on the hydroxide? Attack the carbonyl. Okay, I will attack the carbonyl. All right, what happens when I attack a carbonyl? What must happen? Kick up, kick down. Okay, and then what happens? the bond breaks between the carbonyl carbon and the nitrogen, and it's going to pick up a what? And you have done it completely right. All right? That could have been a test question. Can you do that? Okay. It looked weird, right? There's a lot of stuff sticking out there, but most of that stuff doesn't matter, right? You identify where the reaction happens, that small little area, and you focus on that. All right. Does anybody know what THF is? Tetrahydrofuran, right? If you're an organic chemist, which is a solvent, which looks like this. It is actually the basis of what we call a furanose ring. And the furan ring is what we do. It has nothing to do with what we do in biochemistry. Okay? This is where, we use the same acronyms for two different areas of chemistry. We're going to learn THF in a lot more detail later, but THF is a carbon carrier. Okay? And so you have a hydride carrier? Well, this thing can carry a carbon, but this can carry a carbon as a methyl group. It can carry a carbon as a aldehyde. It can carry just one carbon away from a structure. And it's really strange. And so it's going to take this carbon along with that, what's holding on to it, and take it off. Okay? And so THF is a carbon carrier, and it takes one of those carbons to get glutamate. This is not a reaction that, at this moment in time, I would be able to ask you. But what do we do with glutamate? Yes, into alpha-ketoglutarate and the ammonium that goes into the urea cycle. Okay? Great. Group number 3. All right, what in the world is group number 3? The aromatic amino acids that, except for tryptophan. Okay? So we got phenylalanine and we have tyrosine. All right, phenylalanine's first reaction is to create a hydroxide on it. Okay? Phenylalanine actually converts into tyrosine. Tyrosine then can be degraded further, but tyrosine is a precursor to dopamine. What is dopamine? Do you know? Neurotransmitter. Okay, and so a lot of the neurotransmitters come from this. Melanins, do you know what that is associated with? Skin pigments. And then epinephrine. We've talked about that quite often, right? Epinephrine is a derivative of tyrosine. But in our degradation, we're going to continue to break it down, okay? But let's talk about the enzyme that converts phenylalanine to tyrosine, okay? And some of the reactions. Now, to do that particular reaction, I need a new electron carrier. So the electron carriers we know are NAD plus accepts, FAD accepts, NADPH donates, and then this dihydrobiopterin, okay, is going to be one that can accept, and we got tetrahydrobiopterin and a quinone, and it's a electron acceptor and donator. And what we do is show the reactions with NADPH to show where those hydrides can actually be accepted. And there are two different versions of where the hydrides can be accepted in the molecule. All right? This is the electron acceptor when I degrade. And so I will react molecular oxygen and this particular new hydride acceptor, and I will produce the hydroxide using phenylalanine hydrolase. After I get to the hydrolase, I will use transamination. That is a very long mechanism that uses PLP as a cofactor, which you need to know. and it will convert a single bonded nitrogen into a double bonded oxygen to get hydroxyphenyl pyruvate. These reactions I'm not going to ask you to be able to reproduce, especially when we react molecular oxygen. And so the molecular oxygen reacts, a CO2 is a leaving group, and I get a peracid. That's really reactive. The peracid actually reacts with itself to create an epoxide, and then something really weird happens. What is the strange things that happens? You've learned about hydride shifts, right? You've learned about methyl shifts. But look at that. We shift not just the methyl group, but that whole chain. This is very much a very complicated reaction. Okay? Even this reaction, I'm not going to ask you, but it's very similar to a reaction that you learned in organic chemistry. Okay? Identify for me where the bond breaks. Okay? Is it blue? Is it red? Or is it yellow? Where does the bond break to create the product? Blue, red, or yellow? Sometimes inventory is hard, isn't it? 1. If I break a double bond and I put an oxygen to make a ketone and an oxygen to make a ketone, this oxygen is that one. This oxygen is this one. What reaction breaks a double bond and creates 2 ketones? Ozonolysis, yes. It is not an ozonolysis, but it is similar in outcome. All right. Again, reacting with molecular oxygen is too complicated. This will not be a test question. All right. What is the relationship between these two? Yeah, one cis, the other's trans. It's an isomer. Okay? And so the enzyme that will do this is an isomerase. Okay? And this leads to just about where we need. Okay? Here is a reaction that I might ask you to do on the exam, but what I would give you is this. The other reactant is water. So I give you water. Okay? For that reaction to occur, what would I need to do? What do we do with water all the time? Deprotonate. Okay, that one's easy to do. I will deprotonate. Now I have a hydroxide. Where will it attack? The carbonyl. Will it attack the green one or will it attack the blue one? Green. Electrons will kick up, electrons will kick down, and I will pick up a proton. All right. Question me this. If I put the electron pair on that carbon, is that okay? Is that good, bad, indifferent? Putting a negative charge on a carbon usually is really bad, right? What, putting that negative charge there, what could happen? It could create a resonance structure because it is alpha 2 a carbonyl. So organically it's stable, and so the enzyme is going to use that stability to allow this reaction to occur. All right, this is a test question. What pathway? What pathway? Oh, you know it. Well, at least we covered it. You should know it. Is it glycolysis? No. Is it citric acid cycle? No. Is it pentose phosphate pathway? No. Okay. All right. That's the answer I'm going to put on the test. Okay. Ooh, don't know it. Pentose phosphate pathway. Okay. What else? Did we use that in the urea cycle? No. Gluconeogenesis? No. Your brain runs on what energy molecule? Your heart runs on what energy molecules? What type? Ketone body. And it gets degraded once it reaches the place where we use ketone bodies into acetyl-CoA, which then gets down into the citric acid cycle. No, this one is this is a ketone body. Fumarate is directly into the citric acid cycle. Okay. Now, the reaction that converts phenylalanine to tyrosine, okay, because we have this particular enzyme, tyrosine is not really considered an essential amino acid because as long as we eat phenylalanine, we can synthesize it. But if a person cannot actually cause that reaction to occur because of the hydroxylase being deficient or absent, okay, what happens is I'm going to transaminate, take off the amine off of the phenylalanine. It will give it to pyruvate to make alanine, and I get phenylpyruvate, of which the two breakdown products, phenyl acetate and phenyl acetate, or sorry, phenyl lactate, are both neurotoxins, or they have neurological effects. All right. So what happens if a person lacks this enzyme? One, they have to limit their intake of protein. Okay, they don't want to take in so much protein that they have a lot of degradative processes for the protein. So this upcoming holiday, not great for them because turkey actually has a lot of phenylalanine. So not necessarily something that they want to eat. But what is a tricky thing for a lot of people is who you can avoid protein, but actually if you take aspartame, that is also a molecule as it degrades that creates phenylalanine. And so there is a highlight on anything that contains it that would be a warning sign to those that have this particular absence of this enzyme. And so you need to watch out. So they can't use NutraSweet. They have to go back and use good old sugar. All right. Now, what really happens with those molecules that are synthesized as the end products, mental retardation occurs, and it really affects the myelination of the nerves. And so they become, the reflexes become hyperactive. And so if it is found early enough in their development, you can protect against this. But this is one way in which, if you didn't know about it, this lack of an enzyme can lead to mental retardation. Succinyl-CoA entrance, okay, is methionine, valine, and isoleucine. We're going to create propionyl-CoA and get it to methylmalonyl-CoA. What vitamin is required to convert propionyl-CoA to methylmalonyl-CoA? Vitamin B. Well, since there's more vitamin B's than anything, that is true. But vitamin B, which one? Not 1. Not 3. We can go up the numbers. Not 5. 12. What is the name of the vitamin B12? It's a really tiny, tiny little... It's not thiamine. Cobalamin, remember? We learned cobalamin because we produced propionyl-CoA in odd fats, and it took us that whole radical mechanism to get to the product in terms of our degradative. But This is where we see it more often than the fat entrance. All right. There are lots of steps. 7 here, 6 there, 3 there. Mechanistically, I'm not going to ask you to know anything about this, but you do know that valine, threonine, methionine, and isoleucine will degrade into succinyl-CoA for the majority of their carbons. That's all you need to know for that one. Okay. Group 5, asparagine and aspartate. Okay. Asparagine can be converted into aspartate by the reaction of water. Okay, really, I'm just deaminating the R group. Okay, now aspartate are both of the reactants. Okay, alpha-ketoglutarate comes in, glutamate comes out. What kind of reaction is that? If I go to aspartate to oxaloacetate, alpha-ketoglutarate comes in and becomes glutamate. What reaction that we learned in this chapter, what enzyme would facilitate that? Transaminase. Okay, it's a long stepwise process. Oxaloacetate is part of which cycle? Citric acid cycle. All right. If you use a base, how do you get to fumarate? You have to take off something, right? What do you have to take off? I have to take off the amine. Will the base react with the amine? The base will react with one of these things, right? Are these protons acidic? Yes or no? I said protons next to a carbonyl are acidic, correct? Are these protons sitting next to a carbonyl? No. These protons are sitting next to what? An acid. The reason that protons next to just a carbonyl are acidic is because I can form a resonance structure. But what's already happening to the acid? It's already in resonance, and I can't throw in another pair of electrons just in there. And so they aren't necessarily that acidic. Why do they deprotonate? Because the enzyme is there, and it makes it. And the leaving group would be an A minus NH4. If I gave you that as a test question, I would give you NH4 plus is the leaving group. So I would tell you that what the leaving group is. I will not let you know that there's a base and an acid there because what? There is always a base and an acid there. What does that base and acid actually represent? They are side chains inside the enzyme. So they are specific side chains that one will act as a base and one will act as an acid in the right orientation as part of the facility or how the enzyme facilitates that reaction. Yes?
Would this, because the hydrogens are not acidic, would that require more energy?
It would require the enzyme to stabilize that a lot more. Most likely what will happen is this. It probably has a metal cofactor, okay, that usually is divalent. And what it will do is it will allow the metal cation to coordinate to the negative charge. And if you actually did this, you can create that resonance structure, and what happens is you coordinate both these negative charges to the metal center. And so that's how the enzyme will probably lower that energy state and actually make those hydrogens acidic, even though they're next to that acid. But you wouldn't predict that right off the bat. Okay? All right. So transamination, and we've got that particular reaction. The aspartate can actually also react with arginiosuccinate to create the fumarate in the urea cycle. We saw that reaction, and so that's another way in which degradation can actually happen. Leucine is its own little thing. Okay? This reaction, you should be able to identify that is a transamination. I go from a single bond to a nitrogen to a double bonded oxygen. I will not show the transamination. You can go back and look at it several slides ago. Okay? This one is really similar to a reaction that you have seen before. Okay? You've seen it both in glycolysis, the transition between glycolysis and the citric acid cycle, and in the citric acid cycle. Okay? It is related to how pyruvate becomes the acetyl-CoA, the pyruvate dehydrogenase complex, in which, if it was pyruvate, this would be a methyl group. And then this would be a methyl group. And so it uses the same types of reactions, 3 enzymes that have lipimid in there, FAD, NAD, CoA as cofactors, and do the same reaction. It is complex. I wouldn't actually make you write that whole mechanism out because it's several steps and 3 enzymes. This one would be one I could do on the exam. This would be legitimate. Okay, what do I need to do when I am looking at this? What's the first thing I probably should do? Put everything that's missing, right? And inventory. So I know I have one of those. I know I have one of those and one of those. Do I have any other protons in that reactant? They're all articulated, right? All right. I will articulate how many protons I have in the product. Does that give you a better view of what might happen? Is there an acidic proton? Yes, the one next to the carbonyl. Ooh, what else do I have that helps me in this mechanism? A what? An electron acceptor, right? So a hydride can be part of the mechanism. All right, that's great. Okay. When CO2 reacts with ATP, I get an intermediate, and I showed you what the intermediate is. Ooh. All right. Question. Is this and that methyl equivalent? Okay. One is trans to this and one is cis to that. So not necessarily, although they can react the same. All right. Is this proton or even this proton acidic? Yes or no? Are methyls acidic? No, not normally. Would there be a reason why those methyls, this one or that one, be acidic? It's not alpha-2 a carbonyl. I told you that was one, right? But check this out. If by chance, now don't write this. Well, I'll do it in dotted lines because this is not the mechanism. Dotted, dotted, dotted. Ooh, I can form a resonance structure through that double bond that's connected, conjugated. So I can also do that one with the top. Those methyl groups are acidic. Okay. The mechanism is I deprotonate, then this attacks, I kick up, I kick down, and the phosphate is the leaving group. If this methyl is acidic and that methyl is acidic, why don't I put it there? Why can't it react with this one? Okay, if you're an organic flask, it can. The major product would be here. The minor product would be here because of steric hindrance. Okay? But we're not in an organic flask. The only base, this base is not floating around that can interact with everything. The base only reaches that methyl group. That is the only one that gets deprotonated. That's the only one that can attack. Okay? Beta-methyl glutarol, glucanyl CoA. Whew, that's a mess. Okay, I will add a water. It kind of went around the edge there. And pick up. It just got a little lost. It was following GPS and it went recalculated. Okay. What pathway am I in? Is it glycolysis? Citric acid cycle? Is it pentose phosphate pathway? Urea cycle? Anything else? This is a ketone body intermediate. And my last step is to create the ketone bodies, acetoacetate and acetyl-CoA. Okay? To do that, you have to add a CoA. All right, for next time, I want you to draw and be prepared for this mechanism, add CoA to this, and get to the products. All right? On Thursday, we're going to have a worksheet to work on some of the concepts of the chapters that you're heading up towards the exam. Okay?